Thank you very much, and great pleasure to be here. It's honestly one of the best meetings I've been in this year, anyway, and learned lots. And I hope that we can. What I have to tell you is of interest to you. So I work for a small biotech company that's headquartered in London, UK, and we have offices in Berlin and the US. 100 or so employees, so not at all very large. And our vision really is to transform people's lives around the world using silencing diseases, and that's related to how our drugs work, which I'll explain later on. And and that's using precision medicines to drive positive change to patients, but also to communities around us. So. And what is our molecule based on? It has an sRNA, which is an oligonucleotide. It's synthetic. It's not a biologic or a small molecule, so it's in between. And the important thing is that it's linked to a GalNAc, which gives it specificity to be delivered to the hepatocyte. And that's important because it then hopefully reduces side effects, but it gives you the specificity. We are delivering the drug to the cells that are producing the gene product that we want to interfere with. So I'll advance them all. So what do we hope to gain from these medicines? They're designed to be specific, and I already told you in 2 ways, and to reduce side effects. It has broad utility, so it can be used in rare diseases like PV, which is what we're working on, or in other diseases. We have another product in cardiovascular disease, but they're all related to genetic aspects and non-traditionally druggable products. So meaning you all take aspirin and paracetamol. Those are kind of more traditional ways of doing, and small drugs like that are not usually able to interfere in these more genetic-based diseases. It has a long duration of action, but it's reversible. So it's not a gene therapy. You can take it, and if there are problems, you can stop it, but you only take it very infrequently. And I'll give you an example of that. And we have some other products, as I said. So divesiran is the drug that we're working on, and it's in Polycythemia Vera. And you all know what PV is, but for me, it was more about reminding me that our aim is to control hematocrit in patients with PV and to keep it low, so below 45%. And as you all know, that has the benefit of preventing thrombotic events or cardiovascular events. The drug interacts, is specific for TMPRSS6, which is a regulator of hepcidin. Hepcidin is, that's not me. Okay. Hepcidin is the master iron regulator. So it regulates how iron is distributed in the body. We have data in animal models and also in the first clinical study in healthy volunteers showing that it's safe and tolerated. And those are all published, so if you were interested, you can find the data. But if you're interested and can't find them, just let me know and I'll point you in the right direction. So once the molecule gets into the hepatocyte, it interacts in the cytoplasm, in the cell, with something called RISC, and that essentially breaks down the gene product of TMPRSS6. And that leads to a suppression of the path, it leads to an activation of the pathway that produces hepcidin, and you get more hepcidin in the systemic circulation, which then binds to a receptor in cells called ferroportin, and it prevents the egress or the release of iron. And that's these little silver dots. So when you stop that, you're making iron less available. And in normal people, you need iron to produce red blood cells. In PV, you need a lot of iron to produce red blood cells because that's what the driver is. So by stopping iron, we're stopping that demand for iron, and hopefully leads to reduction of production of red blood cells, reduces hematocrit, and the rest is history. So here, that's what we hope to do: elevate hepcidin, reduce the release of iron from the cells, and prevent iron from entering the bone marrow and reducing red blood cell production. And what happens to that other iron? It gets saved in cells. So what we think happens for fatigue, for instance, is that now cells have a key cofactor to get the mitochondria and all the organelles producing energy so that the cells can do what they need to do. While you have PV, you're starved, all the cells in the body are starved from iron because all the available iron is going to produce red blood cells. So it's changing the balance. It's the whole aim of this drug. So really what we're trying to do is increase natural hepcidin, which is the gatekeeper. And with that, we're hoping to increase iron stores and reduce symptoms while reducing phlebotomies, of course. So that helps us with preventing loss of iron and maintaining the hematocrit to have the clinical benefits that people know about. So Peter always asks me to give a plug for clinical trials. And so drug development is quite a long process. Traditionally, from the lab to approval, on average, 10 years. Sometimes it's 15 and sometimes it's 8. So, and in the case most of you are familiar what happened with, at the time of the pandemic, they produced the vaccines a lot quicker. But that's because everybody was focused on only one thing and not lots of other drugs. So you can do it quicker, but it's not only in extenuating circumstances. So for us, it's taking us about to where we are today, about 8 years to get to a phase 2 clinical trial. And what we're trying to find out really is this drug safe? And does it do what we want it to do in patients? And ultimately, to get it registered, it's not whether it does it or not, it's whether it brings benefit to the patient. That's the key question. So if I'm not doing something for you guys, I'm failing, essentially. And that's why we want clinical trials, because without patients, we can't test whether we're going to bring the benefit. And it's absolutely key. And without patients, we wouldn't be able to do any of the things we do. So the clinical trial that we've been working on recently is a phase 1/2 study. And the phase 1 portion showed that the drug was well tolerated and it had some benefit. And we've just completed enrollment of the phase 2 about a month ago. So we're progressing nicely. Oh, I'm pointing the wrong direction. Yeah?
He's out for lunch.
It isn't.
He's not here, he went for lunch.
Yeah. Yeah. No. Let me try. Okay, I got it working. So what does Enrico stand for? Because you said that's a strange name, and it's from this Latin sanguis for blood and recover for RICO for recover, and it contains RNA in the name, so alluding to our technology. So as I was saying, it's a phase 1/2. It had inclusion criteria, so adults only. We're assessing safety, tolerability, and efficacy. And this is administered subcutaneously, so it's an injection. And we're looking to, of course, understand if there are any safety events, and in the phase 2 in particular, whether we have patients that are phlebotomy-free after treatment. So this data that I'm showing you now is from the phase 1. We only had 21 patients in 3 groups. We were testing 3 different doses. And we gave the drug every 6 weeks for 4 times. And as you can see, we had about 79 phlebotomies in all these patients prior to treatment. And then in the treatment period, only 5. And in the recovery period, another 4. Overall, 9 phlebotomies. So that in part speaks to the long duration of the drug. So you still have a long effect of it. We found the drug to be well tolerated, so very excited. We moved to phase 2. The phase 2 is underway. We've completed recruitment, 48 patients worldwide. And the data, unfortunately, we have to wait till next year to have it. So, but we'll be very happy to disseminate when we have it. And again, the objectives really are about safety, the reduction of phlebotomies, and improving PV-related symptoms. And in the phase 1, we already looked at symptoms because in the original design, we were allowing patients to move from phase 1 to phase 2. So we wanted to have a clean baseline without drug. And so we've already collected some quality of life. And even though there are small numbers, we're seeing that there's a benefit. And so in the phase 2, we hope to really cement that. And so it's very exciting for us, actually. So the study was carried out in 9 countries, 4 continents. And Spain was one of the countries, and we had 5 sites open in Spain, only 2 recruited. And we had Italy, Germany, Bulgaria, Poland is not there, but it was, US, Canada, Australia, and Malaysia. So this is the time when you say thank you very much to all the patients that have participated. And without them, that data that you saw and that you will see next year wouldn't be possible at all. And it's absolutely essential. So what do we do aside from doing clinical trials? We try to support the study with information, videos that show how the drug works, the technology, the drug. They've been translated, or they have subtitles for the different languages in the different countries we were operating in. We produce fact sheets. We produce a PV ebook, which probably every company does too. And but I think the one that I'm kind of more proud of in one way is that we produce a PV book that is a coloring book. And it's about wellness, and a lot of people like to color in, to relax, to kind of escape the gremlins of life. And so this is it. And I'll show you an example in the next slide. But essentially, what we're looking to do in phase 3, for phase 3 as we prepare, is probably update the book, do new materials. And but key is the interaction with patients, patient groups, nurses, doctors, and regulators in the end. And but without all the input, you can't hope to do a study that is going to help people in the end, patients. I don't know everything, and I need the help that I can get from the experts, which are the patients really in my book. The academic experts make sure that we're doing it in a sensible way and that we are following what they know the regulators will accept. But it's the patient that really informs what we need to do. So here's an example of the book. It's, so we had input from different patient groups and patients too. And as a result, this is the second edition. We reduced the format to make it more accessible. We updated, of course, the information. We put patient quotes, but the key there was that we also added kind of mindfulness tips, so to get people to relax. And it'll have, in each page that you turn, it'll have text, quotes, mindfulness, and then a picture to color in. And we also translated into Spanish, so, and the other languages that we operated in. So these are available if anybody wants them. Just email me. It's at the end of the presentation. So looking forward, phase 2 data coming out next year. We'll disseminate, and hopefully we can go to ASH with it, but it'll be late for normal abstracts, so might not. What are we doing for phase 3? We're starting to plan already. So we're kind of 2-3 years before we start. And really, the first thing is talking to experts. Early next year, we'll be holding patient advocacy advisory meetings. So in the US, EU, and APAC. And why that? There's different perceptions by patients, how they feel. They want not necessarily all the same. We heard lots in the last 2 days about how patients feel different, they have different symptoms, they feel different about the disease. We talked to academics, hematologists, clinical nurses, but patient carers and givers, I think, are also quite important. And they are the ones that often are missed out because people don't see them as important, but they actually live with the people that have the disease, and they also need to get something out of this. And ultimately regulators, because we need this to be approved, and so our plans need to be accepted. And we're expecting to start the study in 2 years' time, thereabouts, in 2027. Wow, look at that. That's a surprise. Any questions and comments, please? And while you think about it, those are my details and some websites of interest.
Well, we have a luxury that Dr. Laura Fox is here. She's hidden there in the last row. I've seen that you are having his clinical trial in your hospital. So is there anything you're allowed to tell about this?
Not really, but
Listen, you just
no,
talk.
but we have a very good experience. This trial was very kind in the way that it was open for patients that have certain characteristic of Vera. It was not for all, and we include 2 patients, and it closed well. It's been by the end of October. It was open for a very short time of period because it was full very quick in all the centers, and the experience was really good because this treatment you have to take every 6 weeks, so it's very easy going and very well tolerated. So well, we hope to see the results soon.
Yeah, looking forward.
It's really nice.
And thank you very much for getting us patients. John.
So this was part of my question. You explained how your approach differs from other medications, but more from a technical perspective. I was interested in how does your drug differentiates from other medications if you look at the benefits from the patient view.
In one way, it's too early to say because it's only we've just finished recruiting phase 2, and we'll see the data. But what we were looking for is, can we extend the frequency or make it longer? Meaning, we know that every 6 weeks works. What we want to see is, would every 12 weeks work? And what does work mean? Work means that you give the drug, and for 12 weeks the patient doesn't need it because the hematocrit is well controlled, and they don't have phlebotomies. We are measuring MPN symptoms using the well-known MPN-10, shall we say, and a couple of other things too. But they're all. I mean, it's 48 patients, which is a small study, and it does have a placebo arm. And then once the patients complete the placebo arm, they all move to a treatment, or they all become treated for 3 years. So it has an open label extension that goes for 3 years. So if it's working for a patient, they have 3 more years of it. For the placebo, it's a hard ask because you're saying take it and hope for the best, but for sure after 9 months they'll have the drug. So that was kind of trying to help patients feel that they will get something in the end that hopefully works for them. And in terms of differentiation with other drugs, it's difficult to tell. This drug only really will deal with erythrocytosis, so excess red blood cells. It doesn't do all the other issues that patients have. We don't expect it to change, for instance, the JAK burden, but we're measuring it because you don't know until you know, right? And so we're looking at a few other things that exploratory just to understand better how the drug works. Yes, Laura.
What I find very interesting of this new drug is that, is, as he said, theoretically it won't change the disease, it won't cure the disease, but probably will take out phlebotomies from this, and the I don't know if the word is the blockage, the where it acts, make that the iron that is necessary for other things in the body, not only to get red blood cells. It's necessary for muscles, for concentration, for the immunology. It's important for many things. And with phlebotomy, what we do is we take out the iron, and we are happy because red blood cells are okay, but all the other rest of things are not quite well. So I think it's very important because this is a very different way of thinking of how to treat PV. And it's getting like close to be a direct treatment. So I think it's there are a few treatments going around this kind of way of action. And I think that is very, very important for that. Then we might add to this treatment something to work on the JAK2 buff or to trying to stop the disease. That's another chapter. But if we can take out phlebotomies from this, I think that's very important. And what I think we can say is that when you don't do these phlebotomies, patient feels better. They are not so tired. And I think that's very important, and will improve the quality of life also. And that's the point I see in these treatments.
Okay. Thank you very much.